Okay, so we're now in the last session with our last two talks. And the first speaker is Fred Gould, and we're just delighted to have him here today to talk about applied evolution. Fred got his bachelor's degree in Queens College, and he got his PhD here at Stony Brook. So you're going to see what the proletarian Ivy League can do. And he... there's a challenge for you, and I know he will live up to it. Fred had an NSF postdoc, and then he went to Raleigh, and he's been there ever since, and he's now the Reynolds Professor. He is associated also with the Department of Genetics. He has a long history of interest in agricultural systems and applying the very best of evolutionary biology and population genetics, and also ecologically based genetics, to pest management and pest control. So he's somebody who has been on the front line of applying evolution to agricultural and various applied systems. And he has a large number of publications in this field. He's also been on a number of NRC and NAS committees to try to deal with these problems, which are very important, obviously, in our economy as well as our science. He won the Alexander von Humboldt Prize for his work in agricultural applications in evolution. And what could be more cool than to have the name Humboldt in your vita? So, so without any further ado, Fred's going to talk about applying evolutionary biology, lessons from the past, and challenges for the future.
Great. So Jeff just gave me the challenge here, right? Challenges for the future, challenges for right now. But I have to say it's been a real treat for me, and I really appreciate being invited here because I am usually off on applied problems, and to see the breadth of this has just been great. I've been just trying to soak everything in. So one thing that I heard this morning that was very interesting in terms of talking about applied evolutionary biology, David Hill showed that nice graph of what's happening with the phylogenetics in terms of evolution and how that was low and all of a sudden has this exponential increase. And I don't have the figures here, but I would think if we look at the percentage of people who were doing evolutionary studies who saw application of those studies as an important component, we would have seen bimodality, and that early on and early in the 20th century, you'd see a lot of interest in that, and then it waned, and now it seems to be coming back. So I think that's an interesting thing. And I do want to get through the history of what's happened with that. And so we all know this brave statement of nothing makes sense in biology except in light of evolution. Another brave statement may be that our quality of life depends on a rigorous understanding of evolutionary biology. And so there have been so many contributions in terms of evolutionary biology to our well-being, and some you heard of already, and other people in the audience today, Dan working on Lyme disease and Greg Ray's work with malaria, all are contributions of evolutionary biology to our well-being. Predicting the risk of invasive pests and transgenes all have evolutionary issues. Predicting risks of global warming, all of these come together. Obviously, I'm not going to be able to cover all of this. Instead, I'm going to give you a thumbnail sketch of some small things. And I also want to say that some contributions sort of go unnoticed by the public, but are very important. And in some ways, that's nice when that happens, because some things do get the public attention. And just to give you an example of things that I have been involved in is the use of transgenic crops. And I just want to say, I mean, in some ways, it's sometimes hard to see what is the contribution of genetics versus evolution, or microevolution and macroevolution, but in the end, they come together. But this is transgenic corn next to conventional corn, and this conventional corn has been fed upon by the corn earworm, and that's a huge amount of damage. And there's a toxin that's been transferred here, the genes for it, from a bacteria into corn. And a lot of people don't like to think about those kind of things, but we also have examples of those kind of bacterial genes moving in naturally as well over evolutionary time. But what I wanted to get at is there's a lot of social controversy over this kind of thing, and now we are working with others, including the Gates Foundation, to transform mosquitoes genetically, so that this being the transform one with the genes expressed in the midgut, so that they can't transmit dengue fever and malaria. And what I want to talk to you about is that often public response to one thing affects their assessment of the second. And to deal with these kinds of things historically is important. And when I say lessons from the past in terms of applying evolution, I guess what I want to show is that there are a lot of, I want to give you examples, but also show you the connections and in terms of public response. And actually, you've seen a lot of these faces in other talks, but I'm going to show them to you in somewhat of a different light, because all of these folks were involved in the first applications of evolutionary biology, and that being eugenics. And what I want to show you is a second group, and this is the timeline here in a way that I'll go through, of Vavilov and Lysenko in the Soviet Union in terms of crop breeding and how these things both independently are interesting, but how they interact with each other. And the big thing to notice here is that the issues with Lysenko and Vavilov are thought by many to have resulted in a change here in the increase in wheat yields in the U.S. starting to go up in the 30s, and not until after the 50s starting to go up in the Soviet Union. There's some dispute about that. But what I want to do is get back first to the issues of the first application in eugenics and talk a little bit about that. And here, I think it's been mentioned before that Sir Francis Galton, who was a cousin of Darwin's, wrote a book called Hereditary Genius, basically the self-direction of human evolution, and that was published in 1869, not long after the publication of The Origin. And what is the connection with Darwin? It's a little closer than I had thought before I started looking into this. This is a letter from Darwin to Galton. I do not think I ever in all my life read anything more interesting and original, and how well and clearly you put every point. You have made a convert of an opponent. So there are connections here. Galton worked on this a lot, and both in the political realm as well as in the scientific realm, and wrote maybe the most definitive the definitions and such in 1905, developing the scope and what the aims were going to be of this movement. And it had great impacts. And this is just to give you a sense in the U.S., eugenics at the State Farm. And here's the governor's trophy for the fittest family. And you can see this as something, and it is a little bit funny. I mean, to see these pictures, you wonder. But just down the road from here at Cold Spring Harbor Laboratories on Long Island was the Eugenics Record Office from 1910 to 1940. So it was quite serious in that way. And it was sort of a science. And Pearson, who worked Galton, picked up on this, and he was the founder of the Annals of Eugenics. And this is out of the NC State Library, and you all probably have this, but right, Annals of Eugenics, starting at first volume 1925, 1953, 1954, volume 18, and then it changed its name. And this journal is in existence. It's not one of those very high citation index journals, but it's respectable. And this is the first issue of that, Annal of Eugenics, a journal for the scientific study of racial problems. Pearson was the editor, and here's Charles Darwin quoted right off the bat. And this is the first paper in that journal, The Problem of Alien Immigration into Great Britain, illustrated by examination of Russian and Polish Jewish children. And lots of Pearson product moment correlations in that paper. Another person who figures prominently in here, to my view, is Punnett of the old Punnett Square. And in, I think it's 1918, he published a paper Eliminating Feeble-Mindedness. This is the Journal of Heredity that many of you are familiar with. And he said 10% of American population probably carriers of mental defect. If only those who actually were feeble-minded are dealt with, it will require more than 8,000 years to eliminate the defect, new method, and of procedure needed. So again, this Journal of Heredity. And what's really interesting was this next step was R.A. Fisher's response to that in the Eugenics Review, another eugenics journal, in 1924, the elimination of mental defect. And what's interesting here is he was highly critical of Punnett's assessment, and it's worth reading this. It has been repeatedly brought to my notice in recent months that some calculations published by Professor Punnett 7 years ago have led to a widespread misapprehension of the effectiveness of selection either by segregation or sterilization in purging a population of its feeble-minded strains. The table in which Professor Punnett has inadvertently supplied material for the anti-eugenics propaganda is the following, and he goes on. And it's a very, it's population structure and how to work this out. You could do it in 8 generations, have an impact. So, the bottom line here is that over 60,000 people were sterilized in the United States. So this is, a history of applied evolutionary biology, and as a lesson. So, and Muller was a very interesting character here, who in the 30s wrote this book, Out of the Night, A Biologist's View of the Future. And there's a little thing by Haldane, which in the bottom says, if they are adopted, these practices, the results will be as important as those of the Industrial Revolution. So you can see that people who did a lot of incredible basic evolutionary biology did think that they could have impacts from applying these approaches. And a very interesting thing, in Muller's case, you can see these are his associations, and the bottom here is the Academy of Sciences of the USSR, because that's where he was living. What was interesting in his book was that he had some statements there that indicated it would be good for Russian women to breed with the best of Russian stock to basically the elite to have better offspring and such. And that went over like a lead balloon. And what's interesting is Muller was also a very strong supporter of Vavilov's work in plant breeding. And indeed, Muller left the country, and this was one of the things that was problematic for Vavilov. So what I want to get to now is I sort of gave you a little sketch of the eugenics thing, and now I want to get to this interaction between Lysenko and Vavilov from the early 20s, really through the 60s, but concentrating on this one period. And this is interesting because this is a picture of Vavilov. This is his mugshot when he was sent to prison. And basically there was contention between the two, and Lysenko won out. And there was a lot of work in the scientific community to try to keep Vavilov on top, but did not pay off. And Dobzhansky was very instrumental here, and he actually translated some of Lysenko's work into English just to make sure that everybody could read it and see what trash it was. So over time, of course, Vavilov has been resurrected, and there's a stamp in his honor in 1977. And what is interesting is by 1961, this was a stamp for his brother, who was a physicist. And just 3 years after Vavilov's death in concentration type camp, his brother became the head of the National Academy of Sciences in the Soviet Union. So a lot of interesting things. And I just put a note to myself about dialectical materialism. One of the arguments against evolutionary biology related there, but it didn't seem to impact acceptance of physicists. So what's the deal with these two people? And I just want to go into that a little bit. Vavilov is known for the evolution of crop plants, and I'll show you a little bit about that, Mendelian genetics, pedigree plant breeding, and the building of germplasm banks. so to improve crop breeding. Lysenko came from plant physiology. He coined, I think, the term vernalization in Russian, and did quite a few experiments that are considered to be reasonable about competence to flower based on cold exposure. But of course, his other contributions were Lamarckian evolution and inheritance of graft traits, that traits could be moved across a graft. So, what led to the triumph of Lysenko, and are there lessons to be learned? So this is just my envisionment of the process over time. People talked about having these non-dimensional things. Well, this is prestige. And this is my view of the prestige of Vavilov in the Soviet Union, which around 1917 increased dramatically, went through some ups and downs, and then till his death. And this is what I would picture for Lysenko, and I'm going to explain that a little bit. So Vavilov got a real push when Lenin put on his push for applied science in the around 1920. Vavilov had studied for a few years with William Bateson and came back to the Soviet Union already to get into looking at the evolution of crop plants. And with the support of Lenin, he went all around the world, examined the U.S., South America as well, and found centers of diversity. And this is basically this issue of whether centers of diversity are the centers of origination. And he did an incredible amount of work in this area. And as I said, it was supported by the government. This is the Plant Breeding Institute in St. Petersburg, where they did most of their work once they came back from trips, and that huge staff's working on that. And I think there are legends about people starving during World War II instead of eating the seeds from these collections. He contributed in other ways in terms of evolutionary biology in his law of homologous series, the idea that if he looked at species A and he saw these morphological differences within species, he could predict that he could go to species B, and if he only had this, he would predict he would find that. He had a pretty grandiose kind of view of it, that in some ways he'd be able to do this in the same way that you could have the table of chemicals, that you could predict where a chemical would be. But it's kind of interesting today when we ask these questions about intra versus interspecific variation and where they come from. He was certainly ahead of his time in some ways there. But the main thing was he stuck to his science, and he knew what he could and could not do, and he promised new crop varieties in 10 to 15 years, and there were people who were starving. And he was also having problems growing exotic germplasm in the Soviet Union because of issues in terms of daylight. So what was interesting was at this time, Lysenko arrived on the scene under basically the tutelage of Vavilov, brought in this idea of vernalization, which actually, the idea that you can convert winter wheat to spring wheat just by treating the seeds. But this was great for Vavilov, who was having this problem with his own vernalization of his seeds from other countries. So just because of who Vavilov was, he actually supported Lysenko a lot in early days. So what went on, there's been a lot of histories written about this. The rise and fall of T.D. Lysenko, Lysenko and the tragedy of Soviet science. People bring out these kind of issues, personalities, whether it was Lysenko's personality that got him that way, world politics, very strong ideologies, and starving people. You know, this talk about multidimensional things. Once you get into these kind of histories, I think teasing out what was the main issue, you know, we like to have a simple answer, and it's not. But what I want to emphasize here is the connection between these scientists I just showed you who were dealing with eugenics and what was going on here. And this, there was propaganda on both sides from Vavilov and Lysenko in terms of trying to show themselves as the people who should be followed. So here is Lysenko with old farmers showing them things, and here is Lysenko with peasant women surrounding him. This did not do well compared to Vavilov, who was dealing with this issue. So the politics and the newspapers and all of those things figured in prominently, but I think that these backlogs of how the Soviet Union was responding to the idea of eugenics and how that related to communism was a big thing. And the fact that the more these eugenics movements pushed along and the more prominent Western scientists pushed this, I think this had at least some of an impact on what was going on. And I mentioned Muller. I also want to just point out that Haldane, as much as he was truly an evolutionary biologist, he wrote things supporting Lysenko because of his own political perspectives, I think, at least. I mean, all of this is a bit personal view. But I guess I just want to give you a thumbnail sketch of some of the things going on. I'm not going to give you any huge answers here. But what was really interesting to me, and the reason I don't think that everything is explained so easily, is if you look at Lysenko's rise, there are lots of points where he could have fallen off, but he stuck in there for a long time, and he had this incredible support. Here's Stalin, who helped write some of his speeches. But this was fine in the beginning when he could, pull the wool over people's eyes. But this is a long period of time I just showed you. And this shows some of the failures. And, of course, there's Lysenko. I have him looking over his great field here and his son. So there's a lot of things going on historically. But you can't fool all the people all the time, you would think. But if you look at what was going on with Lysenko over time, even during the era of Khrushchev, and that's the one that I have the hardest time with, is why this maintained itself. And just to comment here, one of the big wins here was in 1948, I think it was, that Stalin outlawed Mendelian genetics. 49, sorry. And not that people in the Soviet Union didn't practice that, but you couldn't publish books and things like that. So this was a huge issue. And trying to understand all the little pieces are hard. But this is Khrushchev again with Lysenko. Think of the President of the United States with some agriculturalists today. I don't know, maybe Obama will get out there. But this was really important to survival. So I just want to paint again this picture and to say that all of these things were going on. But what was going on in these two things I don't think has been explored as much as may be useful. And you might say, like one of these things was about agriculture and the other thing was about people and such. But I want to show you that even in the United States, where the eugenics movement had a lot of followers, the connection between agriculture and eugenics was there. And this was a eugenics poster, connecting seed to that. And here, even more importantly, this is Life magazine in 1946, after Hitler, right? And eugenics in a cornfield. Paul Lombardo lent me this slide. But just to see where these things come and go, it's kind of hard to understand this, I think, in this era. But what I want to do now is shift. How am I doing on time? Okay. What's that? Good, great. This is good timing. Shift from the past to the future. I say lessons for the future. And lessons in terms of if you're going to apply evolutionary biology, what sensitivities must there be? And I can't tell you that I have the answers to this, but at least to tell you a little bit about this. And I told you some about the transgenic and conventional corn and public response to this. Corn features prominently, you noticed in these slides. Okay. But this is not finished. This hasn't played out. And there are a lot of people who have had a response historically to genetic engineering. And even though it's become an environmental issue, that's where the arguments are happening, really it's at a moral stage that can't be argued on global economics and stuff. There's been a lot written about what's going on in the hidden agendas. But what I want to get at is when you have transgenics painted this way because of one move, and I think really imperialism is another big issue here in terms of why there's opposition to all of this, that has sort of gotten people's psyches and lined up in a certain way so that when you now want to use transgenic approaches that really use evolutionary biology to deal with issues like malaria or dengue fever, you have to recognize that you're coming into something that has a legacy. And that's what we are doing. Because what we really want to do is genetically engineer insect pests and indeed rodents as well. But I'll emphasize the mosquito issues here. We want to take a mosquito that transmits malaria or dengue and convert it into a transgenic mosquito that does not do that. And this was the picture I just showed you, was of a normal mosquito and one that has been transformed. The eye is just a reporter, but expresses a specific gene in the midgut. And I'm not going to go through this construct because I'll get myself confused as well, but this is basically an RNAi approach to silencing a gene in the dengue virus when it comes into the insect and only into the female, into her midgut when she takes a blood meal. And this has a promoter, the carboxypeptidase promoter, that is only upregulated in females when they take a blood meal. So it's kind of a cute thing. And the idea is also this is like a very long stretch. So you have lots of small RNA molecules that are interfering. So if the virus adapts to one of them, there's still many more. So there's something to be said here in terms of coming up with an evolutionary stable strategy. But the basic idea is to use that and put that into the mosquito, and people have done that. So far with only dengue virus serotype 2, but more is being done. Okay, but the issue is that's the molecular side to it, so it's not too useful if you think you have a population of mosquitoes of 1 million and you release 1,000 that don't transmit it, you still have the 1 million. So what the evolutionary biologists have been asked to do is come up with this. So that's our approach. We have to come up with a way of moving that into the population. So there have been quite a few things done on this, but I do want to again bring it back a little bit to the history and what's going on. And just to comment that these three people were instrumental in the 40s through the 60s in trying to move these techniques forward, and I'll tell you a little bit about it. But just to emphasize that one of the major people was Serebrovsky in the Soviet Union. And actually, he was in charge of poultry breeding in the Soviet Union. He was a Mendelian geneticist, but didn't do a lot of experiments with these approaches. But basically, what all three of those people were really interested in was to use the idea of underdominance so that they could have, if they were looking at fitness of three, just to make it the simplest possible drawing here, 3 genotypes. You could have one genotype, the normal mosquitoes or whatever it is in the environment, that are more fit than your transgenic one, but if you have underdominance and you release enough of these, you go to fixation of that trait. And if you look at that, and Serebrovsky is very interested in translocations and built models to show what the frequency would have to be when you initially release that translocation in terms of whether it would go to fixation or be lost. And of course, this goes up if there's fitness costs to the translocation. But this didn't go very far, because it was very hard actually to develop translocations that didn't have very large fitness costs when in homozygous form. And the idea was, though, just to back up a little bit, is that you'd have the translocation along very closely linked to genes for not vectoring the disease. Okay? So that when the translocation went to fixation, you'd have hitchhiking of the gene for not vectoring the disease. And actually, Chris Curtis got pretty far with this approach, working in India for about 10 years. And there was an article in the Indian Journal saying that this was going to be biowarfare, that they were just practicing for that with the chemicals they were using. Just to show you that once you take applied evolution and these kind of things out into the public, you have to be ready for things to happen. And that whole program by WHO got shut down. At that time, WHO's policy was never to respond to anything in the press. So what I want to say is that beyond that didn't work very well, but now we're in a different position where I think taking molecular biology and evolutionary biology gives us a lot more tools, all the way from meiotic drive to a killer rescue approach that we've developed. But to make this short, I just want to talk and show you 2 approaches that are being moved along using homing endonuclease and media elements. If you look at all of these, they could all be considered selfish genetic elements, things that drive themselves into the population without benefit to the organism. I just comment on the transposons, which would seem like an ideal one, but the fidelity isn't good enough, so you get too many mutations along the line. People worked on that initially. Just to tell you just quickly about the homing endonucleases. So they're an element that is mostly found in less complex organisms. But if you look at a chromosome with a homing endonuclease gene inserted here, okay, and this is a heterozygote, this is homologous chromosome. So this is the normal homolog, but this is the recognition site for the homing endonuclease. What happens is the homing endonuclease gene makes mRNA, produces homing endonuclease protein that goes back into the nucleus, cuts right at that site, and then you have the repair mechanisms go on, and that homing endonuclease gene gets repaired into the homologous chromosome. So what's interesting is if you have this kind of a situation where you think you're starting out with a homing endonuclease and the heterozygote mating with a normal individual because it's at very low frequency, what's actually happening is that, something skipped here, you're going to actually wind up with F1s with this becomes homozygous during meiosis, right? It converts it, and both copies are heterozygous. So if you look at the gene frequency, it went from 0.25 to 0.5. So you can just imagine something like that in a population increasing very rapidly. So this could be used also if you linked a gene for not transmitting disease to that homing endonuclease, it would move along. But the homing endonuclease could be used in a different way if it was linked to breaking up a recessive lethal. And I won't go into a whole bunch of, I had a lot of detail. Basically, if the heterozygote survived, the homozygote died, you wind up with a situation where you have an early increase over time in the frequency of the homing endonuclease, and then it tails off. I had a bunch of equations that are not really important here. The main thing is that if you had 75% conversion during meiosis, you wind up with a load on the population of about 50%. So half of the eggs, for example, would be dying off. If you could raise that to 90% conversion, you're basically only 1 out of 5 eggs would be surviving, and you could use that for population reduction. The other one in my last minute to tell you about is Medea, and this was originally found in Tribolian beetles, and it's basically that a female that's a heterozygote that has this Medea element produces 2 kinds of embryos, and the one that doesn't inherit that dies, and that's where the Medea name comes from. So you have a heterozygous mother. Basically, the idea is that she lays down an embryonic toxin, doesn't do anything to the initial egg development, but then it's transmitted. You have mating with the father, and once the embryo starts to develop, the ones that had received the Medea element get rid of that toxic compound, the others don't, and they die. And you can see how that would lead to increase in the population. Bruce Hay said, hey, I'm a molecular biologist, I can do this. He built this in Drosophila. He has built a maternal toxin in and a zygotic antidote into a construct, put it in his Drosophila. And this is if the male is the heterozygote, you get great survival. If the female is the heterozygote, you get half survival. They put this in a population cage, and here this shows you the decrease in non-Medea types. So again, another way of moving this in. And I'll end sort of by saying, well, now they're trying to move this into mosquitoes. The Gates Foundation is funding this stuff. We are working in the city of Iquitos in Peru, trying to model the ecological issues here so that we're not dealing with just very vague population genetic stuff. And Matthew Allegro and Alan Lloyd are working on the details of this model that look at specific areas of a city that have been surveyed, look at movement of mosquitoes, and put all that into a model so that we can come up with the best ways of releasing mosquitoes and in a spatial context to find out if this is your city, should you be releasing all the mosquitoes in a very small area, or should you release the same number of mosquitoes more spread out? Or there are a thousand other patterns you could use in terms of how do you establish sort of a Bartonian wave where it would move out into the population. So I'm going to sort of leave it at that by saying that we're moving in this direction, but the question is, how are you become sensitive to doing this in a socially appropriate way, taking into account lots of different cultural perspectives, so that you don't wind up with the response being, hey, doing that. So I just will end by saying that at NC State we've submitted an IGERT proposal, but are already starting to develop a program that has at a core the ecology, molecular biology, and population genetics, but also brings in our colleagues from social sciences and economics. And we're trying to build this so that maybe we can come up with a better way of applying this evolutionary biology. So, thank you.